Hello, everyone, and welcome to today's live broadcast, investigating the locus of conscious sensory experience in the brain, presented by Brian Odegaard, Post-Doctoral Research Scientist, University of California, Los Angeles. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Odegaard. I will now turn the presentation over to him.
Fantastic. Christina, thank you so much for that wonderful introduction. Thanks to everyone at LabRoots for helping organize this event. And thanks to all of you online for taking the time to listen today. So as you just heard, my name is Brian Odegaard, and I currently work as a postdoctoral researcher in the Consciousness and Metacognition laboratory at UCLA. And I also have a secondary affiliation in the Department of Decoded Neurofeedback at the Advanced Telecommunications Research Institute International in Kyoto, Japan. And as you can see from this title slide, what I'm going to talk about today are experiments that I've been doing over the past 3 years to try to investigate which parts of the brain help facilitate our capacity for conscious sensory experience. So in general, as a researcher, my overall goal has been to better understand the neurophysiology and the computational principles that are in play when we look at scenes like this one that you see on the screen, and we have a sensory experience. So I actually snapped this photo when I was back in Tokyo in November. This is the Shibuya station, where I came out after coming out from the airport to try to see some friends in Tokyo. And I thought this scene really captures how incredible our brains are at processing so many different pieces of visual information all at once. So you can see here in this scene of nightlife that there are so many colors and shapes and things happening all at once. And it's really a remarkable capacity that we possess to actually process this scene coherently all at the same time and to understand what it is that we're seeing. So I'm after a more mechanistic understanding of what the brain is actually doing to facilitate our experiences of sensory phenomenology, it's called, our experience of what it actually feels like to experience this visual scene in the moment. Now, this is a question that researchers have been wrestling with for decades in terms of trying to understand which parts of the brain actually facilitate this capacity that we have. And many researchers for the past few decades have talked about how when we think about vision, there are well-established findings in the field that show that what's called the ventral stream is necessary for coding certain visual features that we experience. So as you can see in this image, if you look at the back parts of the brain, the posterior areas that are labeled V1 and V2, there are many neuroscientific investigations that show that these parts of the brain help code for fairly simple visual features. So neurons in these areas areas might be coding for like lines of different orientations, as you can see in the images, or possibly more complex shapes, as you can see in areas like V4 or even parts of the temporal lobe. And we think that there is a chain of processing, a hierarchy involved, where as we move up this chain, more and more complex features are encoded by these visual areas. So a lot of evidence indicates that these posterior parts of the cortex code for visual content. Neurons in these regions help facilitate encoding of visual information. And many researchers subscribe to a school of thought that says, okay, we know that posterior areas are involved in visual processing, but then when we think about what the front parts of the brain are doing, so as you can see in this image, we have parts labeled FR for the frontal regions and PAR, which is actually labeling parts of the parietal lobe, it's called. We can typically think of these areas in the brain as being responsible in facilitating our capacity to report what it is that we're experiencing. So some researchers subscribe to this idea that there's a dichotomy between posterior areas that code for the visual things we see, and then parts of the brain that help us actually speak or report what it is that we're experiencing in a given moment. So this idea has been around for a number of years, and it's actually seen renewed vigor and interest in some recent articles from the past few years, like the one that I'm showing here. So this comes from this image that I'm showing comes from an article by Melanie Boley, Marcello Massimini, Nao Suchiya, Brad Posel, Christoph Koch, and Giulio Tononi. These people are fantastic scientists that have been leaders in the field to try to talk about what conscious experience is and how the brain might facilitate our capacity for it. And this image actually represents this viewpoint that I was just discussing quite well, where if you look at all of the parts that are shown in red in this schematic of the brain, these researchers say that this is really the part of the brain that enables what's called the full neural correlates of consciousness. So you can see that acronym on the slide. It says full NCC off to the right side. And it's this idea that, again, it's primarily posterior areas that help facilitate our ability to experience the sensory world when we're out and moving about. And then you can see with these areas that are shown in black, which are the front parts of the brain, these areas help us do different things, but they're not directly involved in what it is we mean when we say that we're looking for the areas that encode conscious content. So if you take a look at those black areas towards the left side of the slide, there are a few highlighted in different colors. So you can see one of these blue areas. You can see it's important for verbal report. So we know that there are some parts of the frontal lobe that facilitate our capacity for language, and again, being able to speak about what it is that we see in a given moment. You can see the area that's colored in beige in this slide, and they refer to this part as being involved in processing loops. So there's an acronym there that's called DLPFC, and that acronym stands for the dorsal lateral prefrontal cortex. And they say that this part of the brain is very important for processes like attention and working memory. So if we're looking out at the visual world and we want to selectively attend to certain objects, like maybe we want to pay attention to a few people, or maybe there's an animal that's moving, the frontal part of the brain helps us actually select which things we want to selectively attend to in the given moment, but the content would be distinct from this process of attention. But we know that this area that's shown in beige, they say, is also involved in working memory. So if you want to try to store items from, say, a phone number that you've just heard through your auditory system, this frontal part of the brain would help you keep in mind the digits for that phone number for some duration of time. And then finally, you can see in another blue region in the frontal part of the brain, it's labeled M1. This part of the brain is important for motor report. So say you're a scientist and you want to study what people are consciously experiencing in a given moment. Well, one of the best ways we have to do that is simply to ask people what it is they are experiencing in a given moment. And usually, as experimenters, we try to have people push buttons to report what it is that they're experiencing. We sometimes ask for verbal reports. We also ask for button pushes. And according to this group, it's this region of primary motor cortex and a few other areas added in that help people execute these commands to actually report what it is they're experiencing. So overall, this image of the brain is trying to show us that according to this view, we have posterior areas that help code for content, and then parts of the frontal lobe that help code for things like attention and our capacity to report what it is that we experience. Okay, so with that view in mind, part of the goal I have for today is to challenge that view. One of the things that I do as a researcher is to try to study how high-level processes in the brain, high-level perceptual decision-making circuitry that we might think of as being dissociated from these questions about sensory awareness and consciousness, these circuits and these processes actually play integral roles in facilitating our conscious experience of the sensory world. So as an investigator, there are a few different processes that I'm interested in that are sometimes written off as being what's called post-perceptual or maybe high level and being disconnected from sensory experience. And what I'm going to argue today is that some of these processes play an integral role in facilitating sensory experience itself. Okay, so what are some of these processes that I'm referencing here? Well, one of them is the sense of confidence. So if right now you're looking at me or you stare around the room that you're in, you can think about experiencing different aspects of the visual world with different amounts of certainty. But even though you have different amounts of certainty, I'm going to argue that with whatever visual experience is, it inherently involves some degree of confidence in the things that we see. So confidence, or the capacity for metacognition, is a topic that people often attribute to what I mentioned before as being part of post-perceptual processing. But I'm going to show you some experiments I've run today that challenge this viewpoint, and I'll argue for a different perspective, that whenever you experience the visual world, your capacity to have a sense of confidence in what you see or to metacognitively reflect on what you see plays an integral part in experience itself. I'm also pretty interested in perceptual decisions, and my interest in perceptual decisions comes in investigating what peripheral vision is doing at any given moment. So most of your visual field, if you think about what you can see outside of the region that you're looking at right at this moment, it's characterized by this sort of ambiguity. You feel like you experience the surround of the visual field in a high degree of detail, but we know in a number of experiments in carefully controlled environments that you don't actually see the periphery quite as well as you think you do. So part of my research uses a technique, an investigative area called psychophysics, to try to characterize what peripheral vision is doing and how your perceptual system helps facilitates decision making about what it is that you see. So I'm going to argue in a few studies that I'll cover today, you think you see the periphery better than you really do, and once we actually use carefully controlled methods to figure out what's going on, we can actually see how the brain tends to inflate our sense of the periphery. So you think you see more than you actually do, but we need experiments to tease this apart to really understand what's going on with that aspect of conscious sensory experience. And then the other two things I'll cover today, I've done some work on perceptual integration. So one of the things that marks your conscious sensory experience of the world is the fact that it's coherent. You have so many brain regions that are dealing with processing different pieces of sensory information at any given point in time, and yet what's remarkable about your experience as you're watching this presentation, as you're listening to me speak, is that experience is coherent. You don't have the sense that things are disjoint and scattered. I'll talk about more of what integration means later on, but I'm very interested in trying to investigate which parts of the brain facilitate this capacity for coherent sensory experience in the world. And then I'm going to wrap up today by talking about perceptual content. So I also open this talk saying that we think that posterior areas play a critical role in encoding content, those parts of visual cortex that I mentioned before. I'm actually really interested in how prefrontal areas might be encoding perceptual content. So I'll talk some about work that I've done in Japan at my secondary affiliation at ATR to try to investigate how frontal regions in the brain might be encoding the contents of your experience, and see if we can try to come to a new understanding of which parts of the brain you really need to be conscious. So let's dig in. That was a long introduction, but hopefully you see the roadmap now for where I want to go, and we can start talking about some of these experiments and some of the data to learn things along the way. So I want to start today by talking about confidence. Because as I mentioned before, I really think that having some sense of confidence in what it is that you've seen facilitates your capacity for what are called perceptual qualia. This idea that if you're looking at this image that I'm showing on the slide, you can see outside Shibuya Station, there's a Forever 21 sign that's lit up in sort of a bluish, purplish color. You can see some of the other Japanese writing on other signs. And again, if you look at that, you inherently have some degree of confidence in what it is that you're seeing. And this has been an area of active investigation for a few decades now, using non-human primates. So the research groups that have really been pioneers in trying to understand which parts of the brain facilitate our capacity for perceptual confidence have often been studying this in regions that are involved in what's called the oculomotor circuit. So you can see here, there's a picture of a brain, and we have a picture of an eyeball on the left side, and you can see that the eyeball is feeding information in to a part of the thalamus called the LGN. And the thalamus actually connects to many different areas in cortex, but we think that there are a few different regions that are involved in control of how we move our eyes. That's what this circuit is showing here, that also might encode our sense of perceptual confidence. So whenever we talk about brain regions, we always use usual abbreviations and acronyms. And some of the areas that have been implicated in these parts of the brain that help us move our eyes, some of these regions that have been implicated in confidence are the lateral intraparietal area, that's called LIP, shown near the top of the slide, and also what are called the frontal eye fields. So some people that have studied eye movements in this area think there's also a neural signature that encodes our sense of confidence in what it is that we've seen. But recently, I've been in collaboration with other researchers at UCLA to try to study this area that's shown towards the bottom. So it's abbreviated SC, and this is called the superior colliculus. So again, traditionally, the superior colliculus is a region that we know is actively involved in controlling how we move our eyes to different locations across the visual field. There's also some research that shows that the superior colliculus is involved in how we integrate audio-visual information. But we've launched a new investigation to see whether or not the superior colliculus also helps encode our sense of confidence in what it is that we've seen. And this is in collaboration with a few people from Michelle Basso's lab at UCLA that specialize in these types of experiments. So in total here, we can see the oculomotor circuit. We think there are a few different areas involved in confidence, but we're going to focus on the SC, this region towards the bottom here. Okay, so in the first experiment that we launched, the way that people often investigate confidence in non-human primates is with what's called a dot motion task. So the task that we used actually involves two components, which I'll explain here on this slide. So if you look at the top row, what we have trained rhesus macaques to do in this task is to first fixate on a central point. So the macaques are going to stare at a red dot. And then what we will do is we'll show them an array of moving dots. So you'll see in the top row in that third panel, there are a bunch of white dots shown on the slide. And what this is represented is a brief stimulus that we show the monkeys, where most of these dots are moving in completely random directions, but a certain percentage are all moving in one coherent direction. In this example, they'd either be moving to the left or to the right. So we show this stimulus for a brief period of time, and then there's a delay period where the macaque keeps fixating, and then afterwards, when the fixation dot disappears, so you can see that red dot goes missing in the last panel, that's when the macaque has a cue to make a choice by making an eye movement either to the right or the left, depending on which direction they thought the dots were primarily moving. So we train monkeys to do this. They stare at a fixation cross, they see a bunch of dots, most of which are randomly moving, but some are moving in a coherent direction, and then the macaque makes a choice about which direction they think had the dominant motion, to the right or to the left. So you can see in these, in the top row, they have to make a choice to either target towards the right or a target towards the left to report the direction of motion they perceived. But in the bottom row, we also have other trials, they're called, in this experiment, where there's a twist. And you can see in the bottom row, we have a few other targets that are on the screen. You can see one is a green dot, and then there's another gray dot. So on these trials that include this green dot at the bottom, what the macaques can actually do is if they see these dots moving and they're not sure which way the primary dot motion direction was going, if they're not sure if it was to the right or to the left, these macaques can actually, on these trials, select this green option for what's called an opt-out or sure choice reward. So what the monkeys are trying to do here is they're trying to actually earn juice rewards for performing this task. And on these trials, if the monkey sees something that it's not sure of, it can select this green option for a smaller but guaranteed reward. So these trials are interesting because the macaque can either choose to report an answer and try to earn the full reward, or it can select this opt-out option for the guaranteed but smaller reward. And we throw that fourth target that I haven't mentioned yet, the one that's at the top in row B, that is simply to control for lateral interactions between neurons. So if the macaque selects this target, nothing happens. They're not actually supposed to choose this one, and they only choose it on a small number of trials. But really, we're interested in the difference between trials where the macaque makes a choice and trials where the macaque opts out. Okay, so with this paradigm in mind, we then go looking at the behavior to see what this looks like when monkeys are performing the task. And what we have seen is that if you look at this chart on the left side on this slide, we have motion strength, or the percent of coherently moving dots, shown on the bottom axis. And then on the y-axis here, we have the probability of making an opt-out response. So this is referring to what percentage of trials does the macaque actually select that green sure choice option. And what this chart is showing is that on trials that are really difficult, the macaque tends to opt out much more often than trials where it's much easier to tell which direction the dots are moving. So you can see on trials that are easy, where 50% of those dots are moving in a coherent direction, the macaques only opt out a small percentage of the time. But then on trials that are either very difficult or impossible, in the 0% case, the macaques are much more likely to select that opt-out response option for the guaranteed sure choice reward. So this is good. It shows that our experiment is working, that the monkeys are performing in the way that we think they will be. And then we are also very interested in how the difference between those two trial types that I mentioned before on the previous slide between row A and row B, what it looks like in terms of how well the monkeys perform. So what we're showing here in panel D is how often the macaques are correct for these different motion strengths. And then you can see there's two different trial types that we're plotting in panel D. One are trials where the opt-out was available but waived. So the solid gray circles are trials when that green sure choice option was on the screen, but the monkey said, nope, I know what's going on and I'm going to make an answer choice. And we're looking at the difference there between trials where the opt-out was available but waived versus trials where the opt-out was unavailable and the monkey was forced to make a choice. So what you can see here is that on trials where the opt-out was available but waived, macaques tend to get a higher percentage correct than on trials where they have to make a choice to the left or right. So this is very interesting because what this likely means is that macaques are more confident on those trials where the opt-out is available but waived compared to trials where they're less confident, often having a mix of high and low confidence because they're forced to make a choice. So this is good, very interesting behavior. And now we can start asking questions about how neurons might be encoding these differences or facilitating this capacity. So about a decade ago, there was pioneering work done that was published in the journal Science by Rusbakiani and Mike Shadlin that used a similar opt-out dot motion task. And what they found was for monkeys that are performing this opt-out task, they showed firing rate differences for trials where the animal was making a saccade towards the receptive field and trials where the animal was making a saccade away from the receptive field. So there are a few terms I just used that we probably want to unpack to make sense of what's going on. You can see when we talk about making a saccade, that's a word for an eye movement. And when we talk about T in and T out, these are referring to trials where the monkey makes an eye movement towards that shaded gray area that's shown in this panel. And T out means the monkey is selecting the option that's further away from that shaded gray area in the image. So they first show that you can actually look at firing rate differences on correct trials between which target the monkey is selecting. And what they showed in this task that was very, very interesting was that on trials where that sure choice option was waived, so in this example, this would be where the monkey chooses the blue dot, the firing rates of neurons in this part of the brain were at an intermediate level compared to when it selected the other two options. So you can see in panel D here, we have dotted lines that are in between those two solid lines. And what the dotted lines are representing is when the monkey selects that sure choice or opt-out response. So this is very interesting because what we have here are neural firing rate differences between trials where the animal was correct and selected one of those two targets on the right or left. That's the solid black or the solid gray line. And we have those targets where the monkey selected the opt-out, shown by the dotted lines. So this was fascinating, and people, this was purported to be a neural signature of confidence. So we wanted to see whether or not we could find similar neural signatures in the superior colliculus. This is that subcortical region that I showed you in the oculomotor circuit about 10 minutes ago. And what we were intrigued to see was that the firing rate profiles of neurons in the colliculus followed a very similar I should say, profile or style compared to what you see in the LIP. So shown in this right-hand panel for our neurons in the colliculus that we were recording from, you can see that when the animal selects the opt-out, shown by the dotted lines in the image, the firing rates are kind of intermediate. But when the animal selects either of the rewarded targets, either the one towards the receptive field, shown by the black solid line, or the one going away from the receptive field, shown by the gray solid line, there are firing rate differences between those things. So this was fascinating. We have found another region of the brain that looks like it tracks how well animals are performing. But what we wanted to do was go above and beyond these single cell analyses, and we wanted to change the paradigm in a few ways. One is to clean up a potential motor-related confound. So in science, we're always worried about confounds, alternative explanations for what could account for the data that we're seeing. And we wanted to make sure that we could get rid of motor-related confounds that might be present in this paradigm. We're dealing with the oculomotor circuit, and we want to talk about confidence. So how do we know that things are actually tracking confidence and not just motor behaviors? We needed to clean that up. And then we also wanted to look at population level activity. So we know that there are algorithms that do a very good job now of trying to analyze how populations of neurons encode information. And we wanted to try applying some of these algorithms to see if we could discover how the brain is encoding confidence, if at all, in this region. So intriguing first results, but what we next did was we started to build what are called neuronal decoders to try to read out what the population of neurons is doing, which might be linked to confidence. So what we did, we were recording from V-probe electrodes, and usually we recorded from around 8 to 24 neurons on any given day, and we could record the firing rates of all of these neurons across time as the animals did the task. So to decode the information, what we would actually do is take a sum of the spike counts in a given time window, and to build our decoders, we would slide this window across time to read out the neural information. And what this gets us is a sum of spike counts across time for each of the neurons, and we could try to decode information across time to see if we could read out signatures of confidence. So to link this to what I described before, what we really want to do here is look at differences between trials where the opt-out was unavailable and trials where the opt-out was available but waived, because we think that the trials where it wasn't available are marked by lower confidence. We forced the animal to make a choice, and trials where the opt-out is available but waived are marked by higher confidence, because the animal could have selected that sure choice option, but it didn't. So we think this comparison in this experiment would be informative for trying to read out a signature of confidence. So again, we want to compare what I mentioned before were target in and target out correct trials as a function of opt-out availability, following the logic that I just described. So again, we're looking for a neural signature that can distinguish between those solid gray circles, those trials, and what neurons are doing on the trials where there were those white open circles. We want to see if we can read out the difference in what neurons are doing between those two trial types. Okay, and what did we see? So what I'm going to show here are results from our decoder. And as we were trying to read out signatures of confidence, the first thing that we did was we tried to decode what the animal was going to do when the answer choices popped up on the screen. So ideally, if things are going the way we think they are, we would not be able to decode what the animal is going to do in the end until it's actually seen the dots. Supposedly, as the animal sees the dots, presumably, the neurons are going to be encoding this information, and the animal will accumulate evidence and eventually come to a decision with some degree of confidence. But we first wanted to see whether or not we could predict the choice that the animal was going to make. So to make sense of this plot that I'm going to show, the y-axis here that's labeled posterior probability, we are trying to actually predict whether or not a given trial that we're trying to decode involves a correct T-in or T-out choice. So the y-axis here is the probability of predicting a correct T-in choice. And to make sense of what we're actually showing here, you can see that when the motion signals come on, something interesting happens. So on trials where the opt-out was available but waived, these are shown by the blue lines. And you can see that after the animal starts seeing the dot motion and it starts integrating evidence and coming to a conclusion about what choice it's going to make, then if we're decoding from these neurons, we can actually predict the choice that the animal is going to make. So what we're showing here in these blue lines is the neural firing rate differences allow us to decode on a given trial whether or not a T-in or T-out choice is going to be made. So these blue lines are showing those trials where the opt-out is available but waived. And you can see that the difference on these trials where the opt-out is available but waived is greater than the trials where the opt-out is unavailable. So this comparison is very, very interesting. We wanted to find a neural signature of confidence. And here we can look at how neurons are integrating information across time, read out the population level activity, and you can see that we have firing rate differences in the population that are informative. We have differences between trials where the opt-out is available but waived, shown in blue, compared to when the opt-out is unavailable. And on the right-hand side of this slide, you can see that by the time an animal makes a saccade, when it finally makes the choice, then we can decode with almost perfect accuracy what's going to happen. But it's really this intermediate range in the middle that's informative. We see firing rate differences, which seem to track the differences between these trial types. Okay, so that's great. For most circumstances, we know that typically in the world, as you are looking at things, how confident you are tends to track how accurate you are. So usually, if you're able to do a perceptual task and do it well, you are going to be more and more confident. But one confusion that people often have is they often think that confidence is the same thing as the probability of how accurate you are in a task. And these things actually dissociate in very interesting ways. So capitalizing on a paradigm that was introduced in the psychophysics literature a few years ago, we wanted to come up with a paradigm to dissociate your sense of confidence from how well you can perform the task. Because in science, we often want to try to isolate these variables to find a true neural signature of something. So what we wanted to do in this second experiment is to come up with a paradigm where the task performance would be the same across two conditions, but we'd get different levels of confidence in terms of the reports we get from the macaques. So the way that we did that, shown in panel A, is we used our same dot motion task. We're still going to see stimuli with a bunch of randomly moving dots, but now we're going to change the task just a little bit so that we will compare conditions where you have a lot of dots moving in the coherent correct direction, but then some percentage of dots moving in the incorrect direction. So the task for the monkeys here is still to report the dominant dot motion direction, but what they're going to have to do is they're going to have to figure out what's going on across two conditions where the ratio of dots moving in the correct and incorrect direction is the same. And what's very interesting about these two conditions is you can actually match task performance shown in the light gray bars in panel B. So you can have roughly the same percentage correct, we'll say, across the two conditions, but these conditions produce different degrees of confidence as indexed by that opt-out response, how often they select the sure choice option. So again, here what we're showing is between these two evidence that we call low positive evidence and high positive evidence, you can see that these dot motion ratios produce the same level of performance, but as shown by the dark gray bars, they opt out much more often in one condition, the low positive evidence condition, compared to the other. So this is interesting. We have conditions that now produce the same level of accuracy with different degrees of confidence. So now we can go look in the colliculus and see whether or not we can find neural signals which track that. Okay, and what did we see? Well, it was interesting because the effects that we saw in the first experiment went away. So when we looked at these differences between T-in and T-out trials across these two new conditions, the high positive evidence condition and the low positive evidence condition, so those are the blue and the green lines respectively, we actually couldn't decode the same differences between those two conditions as we could in the first experiment. So you can still read out T-in versus T-out differences, but there's nothing in the colliculus that seems to distinguish between those two conditions. So that's a lot to digest in terms of its relevance for the topic, but let me summarize here what we have learned from these two experiments. So first, conceptually, it's a little bit hard to wrap your mind around at first, but confidence is different from task accuracy. Yes, those two things normally go hand in hand, and people have been saying that and coming up with statistical frameworks to formulate that in quantitative and rigorous ways, like the paper I'm citing here. But these really are two different things, and we can come up with clever paradigms to try to exploit that to find the neural signatures of confidence in the brain. We saw in the first experiment that the colliculus encodes a form of confidence that's similar to other parts of the oculomotor circuit, like LIP. So the colliculus tracks task accuracy, which is very interesting. This is something that we didn't know that it could do effectively. But we don't think the colliculus encodes true subjective confidence. So in terms of trying to identify the part of the brain that facilitates our sense of confidence, we don't think that that true sense is in the colliculus, nor do we think it's in some of these other regions that have previously been reported as having the neural correlates of confidence. So what this really has led to is we've surveyed the literature, and we think we have here a paradigm which others can exploit to investigate further which brain regions really encode that true sense of confidence. And we know from the human neuroimaging literature that prefrontal cortex is likely involved in that process. There are many, many studies that use what's called fMRI, which show that frontal parts of the brain activity in those areas seems to be linked to our sense of confidence. And we're following up on this study in the colliculus to try to see if we can see that in this investigation as well. Maybe neurons in prefrontal areas will encode the true sense of confidence, since we've now been able to rule out true subjective confidence being in the colliculus, or at least we have null results that provides evidence for that. And we think these results challenge the conception of it being in other areas too. Okay, so that's a lot to digest. That was actually the biggest thing that I had to cover today. Hopefully the rest of the topics will be a little bit easier to cover. So we've looked at confidence, we've challenged some ideas about where that resides, and we know that there's some evidence that's in frontal areas. But what about perceptual decisions? I mentioned originally that my interest in perceptual decisions comes mainly from my interest in peripheral vision. And peripheral vision is weird. So if we look at what peripheral vision is like, there's this image I like from a recent paper by Michael Cohen and Nancy Kanwisher. And for those of you, you might know the name in philosophy, Dan Dennett. He was an author on this paper as well. They've argued that when you think about what peripheral vision is like, it's surprising. We know that the number of photoreceptors and the resolution tails off outside of what's called the fovea, the central region of visual space. But they note in this paper that when you think about what peripheral vision is like, the world doesn't fade to black outside of where we're directly looking. It looks roughly like some sort of smeared, rough, summary representation, like the panels that are shown on the right-hand side of this image. So again, peripheral vision, it doesn't all fade to black. There's some degree of resolution. There's some information that we have, but it also looks kind of smeary and blurred, and it can be tough to characterize what's going on. So my work has tried to build on some interesting findings from the last few years about what characterizes peripheral vision. So one of the findings that caught my eye was work by Guillermo Solove and a few other co-authors that showed that in the periphery, when we make judgments about what it is that's in the periphery, we tend to have liberal response biases. So if we are looking at tasks where you simply have to detect whether or not, say, a simple visual grading was presented, we know that people tend to report the grading being present much more often than it actually was. We tend to have liberal detection biases outside the center of our visual field. So that's what this chart is showing right here. We can measure response bias and show that people tend to have liberal detection biases in the periphery. And this has been shown in other work as well, where if we look at how people perform in tasks where they are either attending or not attending to certain regions, that's the cued and the uncued conditions, we know that in areas where you're not attending, these liberal response biases, shown by the parts labeled C here, they tend to show up. So if you're not attending or if it's in the periphery, you tend to think you see things even when they aren't there. And I've started some collaborations. I have a fantastic co-author. His name is Mitsuo Lee from Tsinghua University in China. And Mitsuo has coded up new tasks in driving simulations. So he actually has coded some things that look like virtual reality. And we've conducted experiments where people drive down suburban roads, and we ask them what they actually see in the periphery as they come up to stoplights. And we ask them about the color of pedestrians' clothing. So as people do these tasks, we have them make judgments about people that are presented in the periphery of their visual field in the simulated driving environment, and we simply ask them whether or not the person was wearing a yellow shirt in the experiment I'm going to show results from right now. And what we've seen in the data is something interesting, where let's just look at panel B here for the sake of simplicity. On trials where people are attending to the location that I'll ask about, where I'm going to ask you whether or not a person was wearing a yellow shirt, and in the valid condition that's shown in panel B, you know that ahead of time. You know where the person is going to show up, where you'll have to make the shirt-related judgment. What we have shown is that people are pretty accurate in that condition where you're attending to where you should be. But if we compare that to conditions where you're not attending, that's the invalid bar that's shown in white right here, you actually have more liberal response biases. So in this experiment, you are much more likely to say, yes, someone with a yellow shirt was there, even when they weren't. So we also have seen in these naturalistic environments that you tend to be more prone to false alarms, they're called. You tend to say that you think you saw things that weren't actually there, and that's pretty interesting. We've also done this with more complex stimuli. So I have started using Amazon MTurk to try to bring visual experiments to the masses recently online. And I've run some tasks where I have people look at a briefly flashed grid of lines of different orientations. And in one online experiment that I run, I flashed these lines either in the center of their computer screen or in the periphery. And I simply asked people, was there a group of lines that were all tilted in the same direction? So I'm just going to flash some lines, tell me whether or not there was a group of lines that are all tilted in the same way, and then tell me how confident you are in that response. And what we've seen in this type of task is that shown on the left-hand side here, people tend to be slightly better doing this task at the center compared to the periphery. So that's not surprising, right? When we try to make visual judgments, we would expect to be better when we're doing it right where we're looking compared to doing it off to the side of our visual field. But what's interesting, again, is that even though you're worse doing this task in the periphery, you can see in the dark shaded bars on the right-hand side, you once again have a more liberal bias to say that, yes, there were coherent lines there, even when they weren't. So again, yes, you're worse in the edges of the visual field, that we would expect. But if I look at the way that you're wrong, you're often saying that, yes, that thing was present, even when it wasn't. And we've seen this phenomenon show up in other ways. So some of our collaborators in Hong Kong, including Xingheng Chung and also Minyu Chang, they conducted an investigation using a phenomenon called crowding. So you can see in this experiment, if you ask people to judge the tilt of a grating that's right above this white fixation cross, it's hard to do when it's surrounded by other items. So you can see in the second panel here where it says or, so those two boxes to the left and right, when you only see one thing on a screen, it's pretty easy to tell whether or not this thing is tilted to the left or right. But when you have three things that are bunched together and you have to judge the middle one, whether it's to the left or right, that's actually pretty hard. So this is a phenomenon called crowding, where to identify something that's crowded by other objects in your periphery, we know that your sensitivity and ability to do that kind of tapers off over time. So we've looked at people's performance on these single and crowded conditions, and we also asked them to rate confidence in their responses. And what they showed in their study from Hong Kong University was that, as you might expect, shown on the left-hand slide here, people are worse in the crowded conditions compared to the single conditions. So that's not surprising. But what is surprising is the fact that your ability for your confidence judgments to track how accurate you are is impaired in the periphery. So I have more jargon than I have to throw at you. There is a measure that we use called the M ratio. So what is shown in the dark gray bars here is a measure of how well your confidence judgments track whether or not you're right or wrong. So that's what the dark gray bars showed. And if you are optimal, if your confidence judgments when you're not very confident are often like wrong, but when you're highly confident, you're often right. If you're doing the task optimally, that measure will be right around 1, which is what you see in the single condition in the dark gray bars. But we know that the ability of confidence judgments to track whether or not you're right or wrong, it tapers off in the periphery. So you can see that that measure in the dark gray bar for the crowded condition is below 1. So that's interesting. Your ability for metacognitive reflection on what it is that you see in the periphery is impaired. Okay, so we've learned things that are interesting from these psychophysical investigations. Psychophysical investigations indicate that subjects are more likely to say certain visual attributes are present in the periphery even when they're not. So you have that bias towards false alarms that I talked about. You think you see more in the periphery than what's actually there. And then finally, we know that you have impaired metacognition for crowded items in the periphery. So the periphery is always cluttered. You always have so many things that are off to the side that are difficult to identify. And it's interesting that your ability to introspect and reflect on that is impaired compared to the stuff that you see when you're directly looking at it. So that's interesting. That's work that we have under review. It should come out in probably a couple months. And we're very curious to hear reactions to that, because I think it's an interesting finding that, again, shows us that we think we see more of the periphery than we really do. So a couple other things I want to cover. I have also been interested, as I mentioned before, in perceptual integration. So let's say you're at a cocktail party and you're working your way around the room and you have to try to figure out, it can be loud and it can be crowded when you're in those noisy environments, whatever, like weddings or bars or wherever you're at, and you're trying to understand what people are saying. Your brain has to do a really good job in those types of social situations to figure out who it is that's speaking, like which things that you're hearing actually go with which voices. And for those of you that have experience in social situations, you know that when you're in a really crowded bar at a wedding and it keeps getting louder and louder, it can be harder to make those perceptual inferences about how to bind voices with who is saying what. And I've been interested in that problem as a neuroscientist, but the way I tried to address integration in controlled scientific settings is to use simple settings that test our perception in space and time. So the way these investigations normally work is that people in my spatial tasks will try to localize where I play briefly presented flashes of light and bursts of sound. So typically in these tasks, I'll play simultaneous bursts of sound and flashes of light, and people have to tell me where both things occurred. And we know, based on this effect that's called the ventriloquist effect, that where you localize sounds are often pulled towards where you see flashes of light. So we know that the way the brain binds things in space is it often, there's a degree to which it's called cue-related, cue reliability weighting. I won't get into what that term means. It's a little too complex for the time that we have left, but we know that typically your sense of vision is best in the spatial domain. So where you think you heard things get pulled towards where you think you saw things. And in time, it's actually the opposite. So if you look at this panel on the right, if you play visual and auditory things that are separated in time, we know that your sense of hearing is a little bit better in temporal perception than your sense of vision. So the way that this typically works in most circumstances is that your sense of when you think you saw something can be modified by when you heard something. So we know that your visual and your auditory senses interact in different ways. And some very smart people about a decade ago came up with computational models to try to account for the quantitative principles that the brain might be using to make these judgments. So my work focused on using these models, which I'm showing a graphical representation of the model here, which says that whenever you encounter audiovisual signals, your brain has to decide whether or not those signals labeled XA and XB come from the same source and should be integrated, or come from different sources and should be segregated. And the bottom line is that there are complicated rules and interesting math that's involved to account for how we deal with this. But my research was interested on the perceptual prior that's shown on the right-hand side of this equation. We know, you might have heard if you listen to Anil Seth's TED Talk or other perceptual researchers, that perception involves inference about, it's essentially the brain's best guess at what's out in the world. And we can use mathematical rules like Bayesian statistics to try to talk about how whatever it is we experience in a given moment is a combination of not only what we see, but these pre-existing biases we have to interpret the world that are called priors. So one famous prior that you can Google is called the light from above prior. If you look at ambiguously shaded scenes, there are ways that we normally interpret shading based on this prior that we have built into our system. And I wanted to investigate this prior for integration, because it seems like this plays a critical role in how we have coherent perception of the world. So what I showed through a number of studies was that these priors are actually stable over time. So if people do these tasks that I just mentioned in different sessions, they tend to have similar values for this binding prior, but these priors tend to be task specific. So it's not as though we have one global prior in the brain that seems to influence how we bind things in space and time. It's more like we have different priors for different perceptual tasks that influence how we perform. So shown here is the stability of that prior, but then if you look at how it generalizes between those two tasks that I mentioned, the temporal task and the spatial task, anytime in statistics, if you see a cloud of dots like that, usually you can be pretty sure that there's no relationship between those two things. So that's evidence that the prior is actually fairly task specific. I was also very interested in whether or not these priors could change, and I had some results that show that if you flash things that are far away from one another in that spatial task over and over and over again, that's shown in the right-hand panel, you can actually increase the prior in people, so they bind more often. So that was some work that I did a couple of years ago that showed that you can increase the spatial prior. I have some modeling results that I'm just showing on this slide that you can pause and look at later on to show that you can actually increase this prior in most subjects, going from a pre-test to a post-test phase. And then finally, we were interested in whether or not that prior relates to schizophrenia. So if you think about the symptoms that characterize schizophrenia, one of the things that characterizes perceptual experience is the fact that in schizophrenia, there's sort of a disjointed nature. People have impaired perceptual inferences. And we saw that this binding prior is at least moderately related to the number of prodromal features, they're called, in schizophrenia. So for people that have more abnormal sensory experiences, we show that their binding prior tends to be lower. So it's an interesting relationship that we showed, but I want to follow up on that. So it's not overly strong, as you can see by the data here, but we think it'd be interesting to try to see if in clinical patients with schizophrenia, is there actually a stronger relationship than what we showed here with people from the general population. So I'm going to skip my summary slide here in 36. We have used these models in many different ways, so I can put up the list of all the publications of how we've applied Bayesian causal inference models to different problems. I mentioned Anil Seth's TED Talk, so if you're interested more in perception as a process of inference, to hear more about that in a way that's accessible for the general public, I recommend you listen to Anil Seth's TED Talk on the TED website or on YouTube, wherever you can find it. And if you're interested in these publications of applications of Bayesian causal inference models, I have that list right here. But I'd like to wrap up with one more thing that I've been involved with today. And that last topic that I mentioned before was, where does perceptual content reside in the brain? We've been very interested in this question as a laboratory, and we have launched a massive neuroimaging project to try to decode which parts of the brain are involved in how we perceive certain categories. So in the work that I've been doing at ATR in the last year, we actually have a database now of more than 50 subjects where people lie in a neuroimaging scanner, and we show them 3,600 images from 40 categories. So in these experiments, everyone is lying in the scanner and they're seeing a sequence of about 1 image per second from each of these 40 categories, and there are 3,600 images in total. And this is work that actually was started 2 to 3 years ago from 2 guys in my laboratory, Vincent Taschereau de Michel, a fantastic postdoc that I've been working with. He just published an article in PNAS that relates to the work I'm going to describe here. So you should Google him, check out his article. And also we have J.D. Knotts, who's a grad student in our lab, who has other work that he will be publishing in the coming year that also stems from this project. And so I wanted to give them credit before I talk about this, because I have learned a lot from them, and they helped get some of this work off the ground. But as I mentioned before, we're really interested in how these 2 parts of the brain, which we call DLPFC and VT, might encode these images that people are seeing. Which region is really critical and has encoded the content that people are seeing. There's a lot of debate right now in the scientific literature about when we try to decode information from different regions, is it actually just plain harder to decode from DLPFC because neurons encode things in more complex ways? So there's a great article up on bioRxiv by Bandari and a few other authors. I think David Badre is the senior author there. And they say, look, we know that in neuroscience, we've had success decoding from things like occipital and ventral temporal areas, but if you look at the results in the neuroimaging literature, decoding from frontal cortex is flat out hard. But we have tried to get around this problem of the difficulty of decoding from PFC by applying what's called hyperalignment. So there's an effective technique for trying to align brains across participants to more effectively read out the content in different regions. And I know I'm running a little low on time, so I won't get into all of the nitty-gritty details on what hyperalignment is and how it works. But we have found fairly effective ways to decode from PFC. What I'm showing here are just some pairwise classification results for trying to decode whether or not people saw one category versus another category. So if I want to know at any given point in time whether or not someone is seeing, say, a snake versus a rabbit, or a dolphin versus a bat, if those are the 2 images I'm interested in, I found early on that you could actually decode with a very high degree of accuracy from parts of frontal cortex. So these accuracies that I'm showing here are committing a giant sin in machine learning. They're both training and testing on the same data set, and there are reasons why I'm showing this figure here for the project I did and how we built decoders. Don't believe the y-axis on this slide, but know that the content is there. We have done analyses that are much less sinful and much more correct in terms of trying to decode information. And we saw early on in this box plot that I'm showing here that you can decode the category that people are seeing, whether or not you're decoding from VT, those posterior areas, or PFC, which is shown on the right-hand slide. So early on, we saw that we can decode content from both the posterior part of the brain and the frontal part of the brain. And I'm very excited to say that in the last few weeks, I'm just going to give a teaser here, and I won't actually show the data. We have been able to blow these decoding accuracies out of the water in both of these regions using hyperalignment and trying to come up with better algorithms to read out the perceptual content. So you can see on this slide from our early results, we can decode above chance in PFC. And I can tell you from the last 3 weeks, we are able to decode with a very high degree of accuracy from both of these areas using hyperalignment. So I have to end the main scientific part of this talk with just a teaser, a little bit of data here, and then a teaser. We are going to be talking about these new techniques and these new results at the ASSC conference in Poland in the coming year. And I should also be presenting this later at the Society for Neuroscience conference in 2018 in San Diego. So for those of you that are skeptical that prefrontal cortex encodes perceptual content, we have very interesting results to show you that go even above and beyond the data that I'm showing you today, which shows that we can decode things above chance. So there's ways that we've tried to build on hyperalignment and deep learning and a number of new techniques in the field that have worked very well. So let's take stock as this comes to a close. You have seen today, or at least heard from me, and hopefully I've convinced you, that sensory awareness and your conscious sensory experience involves confidence, decisions, integration, content, so many different things that all make up what it means to have an experience. And I think today we've seen that some of these ideas that we can dichotomize the brain in certain ways to talk about where content resides and where essentially processes of report and attention reside, that might be oversimplifying things. I'm not denying that the giant areas in red shown here in this image are important for perceptual experience. I think there's a tremendous amount of scientific evidence that indicates that yes, that is the case, but I would argue that with a number of the findings I showed today and some of the findings that I referenced as well, there is increasing evidence for a role of frontal cortical areas in facilitating conscious sensory experience. And for those of you that are interested, I'll close by showing just a couple images. This has been a very interesting debate we've been involved with that was published in the Journal of Neuroscience. So if you're interested, you can look up these articles by Melanie Boley and her co-authors, as well as myself, and watch some videos online of patients that have different types of brain lesions. So sometimes you can see in patients really vivid examples of how sensory experience or function might change based on where the lesion is. So I recommend checking out our discussions of not only historical data, but also more contemporary patients. And there are some other pieces of evidence, like the one I'm showing here, that if you dig into that article in the Journal of Neuroscience, you might find helpful. So just to conclude today, when you think about what conscious experience is like, one of the metaphors that people have used in literature is the Cartesian theater. So it's almost like when we look out at the world, there's a little man inside our head that seems to have access to like a movie screen, the way the world is rendered in front of us. And my argument today would be that when we think about which parts of the brain facilitate the content that's on that movie screen when we look out at the world, I know that posterior areas do play a critical role in facilitating the content that's on the screen, but I would argue that you don't get a seat in the Cartesian theater unless you have your frontal lobes working, and specifically the dorsolateral parts of your frontal lobe. So it's a fun debate that we're involved in. We were using a lot of paradigms to try to investigate this topic, and I'm excited to see what results people come up with in the coming years to try to resolve this debate about which parts of the brain are necessary in getting us that seat in the Cartesian theater. So I'll close today before we turn it over to the Q&A with just acknowledgments of a number of people that have helped with this work in the last 5 years. Thanks to my postdoc advisor, Hakwan Lau. Michelle Basso's lab facilitated the early experiments that I talked about in this presentation. Bob Knight I had great discussions with him about that Journal of Neuroscience article I referenced. And I probably can't go through everyone else on this slide in the way that I should in the time that I have left. But everyone that is listed here, know that I am extremely thankful for what it is that you contributed to all of these projects. It would not have been possible without all of their help, including all of the names I have wedged in the lower right-hand side of this slide. I did my graduate work at UCLA and had a fantastic army of undergraduates to help with everything. So for even the small names that are towards the lower right, thank you to all of my undergrads that help with these projects, to all of my mentors and collaborators listed on the slide. And if you made it this far online, thank you so much for listening as well. So I'll turn it back now over to Tiffany, and hopefully we can have a brief Q&A as we wrap things up here. So thanks again to all of you online, and thank you to all of you at LabRoots for organizing this.
Well, thank you, Dr. Odegaard, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We will answer as many questions as we have time for today. So let's get started with our first question. And this question asks, why would our perceptual systems have a tendency towards making false alarms in our periphery for object detection?
So this is a great question. Normally when we think about our perceptual systems, we think of these in the context of evolution, right? So evolution over a period of hundreds of thousands, if not millions of years, has been sort of guiding how our perceptual systems work. And typically we think they're going to be getting better and better at making use of information in the sensory world around us. So this experimental evidence that we have for why we tend to make false alarms seems counterintuitive. You would think that our perceptual systems would be close to optimal with how they make use of information, and they're not. So that's a good question. At this point, all we can do is speculate, but I want to actually reference a researcher by the name of Michael Graziano here, who published a piece in The Atlantic wrestling with why we might see some of these interesting results when evolution should have weeded them out. And his idea was there might be times where if we're trying to survive in a dynamic and changing world, that our perceptual systems might be prone to sort of have a trigger finger, if you will. They might be prone to jump the gun to try to say that something is there, even when it's not, to spur action. So this could occur in the context of like predator identification. So our perceptual systems might have a bias to try to think things are out there even when they're not, because that could be optimal in motivating our either approach or avoidance of things that might be dangerous. And similarly for things like food or mates, having a little bit of a tendency to jump the gun and think something is there might spur action in a way that actually could be optimal to enhance survival rather than simply remaining stationary until we have more information. So I would say at this point, we're still only speculating about the evolutionary reasons why that might be in place, but we're going to try to conduct more experiments in our laboratory to probe this question more thoroughly to see how widely this tendency for false alarms generalizes across different contexts and as we ask different types of questions. So it's a good one. We have ideas for why evolution might have put that in place, but we need more data before we can say something conclusive about it.
Excellent. Thank you so much for your answer. The next question asks, where do perceptual priors come from, and are they established early in life through sensory experience driven by genetics or some combination of the two?
This is another good, difficult question where there's not a true consensus about the answer. So I mentioned before that light from above prior. And for those of you that are listening online to this, I actually recommend that you Google that right now, and there should be a couple images that pop up that show you vivid demonstrations of the light from above prior. And this has been an open question about why that prior might be there. Is it something where when we're little kids, we always experience lights from above on the ceilings and we know the sun is above, so maybe we have this tendency to think light comes from above just because that's the way it always happens in our world? Or other people think that, you know, as we have evolved over the course of many, many, many years, maybe that's actually been built into our systems inherently to interpret the perceptual world in this way. And I had a conversation with someone who claims, I'll reference this, Brian Shaw from Yale University told me this story about he was, as a graduate student, digging in the library in the stacks one night looking for another article, and he told me he came across this interesting experiment where they raised chickens in the 60s from an environment where the lights always came from below. So someone about 40 or 50 years ago actually tried to conduct an experiment with baby chicks, and they asked, okay, if I always raise animals in a way where the light comes from the other direction, and I show them these ambiguous stimuli where we usually see, because of the light from above prior, things are indented in certain ways. He said that in this article, they showed that even when the light always came from below, they still seem to have a light from above prior. So this is actually one example that it seems as though even if you change someone's life experience or you change the life experience of an animal, it still seems like the light from above prior is still present. So I would love to see that old study replicated in some ways, because I think it would be fascinating to have more modern data to speak to this question. But I actually come from the stance of I think it's actually something that now is built in genetics. There's no doubt that priors can change based on sensory experience, but it's a question to how much they can change. And right now, I tend to think that our perceptual systems tend to have a way of being wired that lead us towards these biases. But that's another question where we need more data to answer it conclusively. But it's a good one.
Well, excellent. Thank you once again. Our last question of the day asks, what are future directions for your research on prefrontal cortex now that you have shown you can decode object categories effectively from this area using fMRI?
Excited about of all of the things I'm involved with right now in research. There are so many times we debate like what consciousness is, that it's such a fun topic, but it never really feels like we pin things down conclusively. So like everyone from philosophers to neuroscientists to lay people, it's such a cool question about what is consciousness. And now that we have better techniques in fMRI, I really want to do two things. So one is to try to read out more vivid types of perceptual content. So if you take, say, the visual domain and you break it up into things like hue, which is a sort of a synonym for color, or saturation, or value. You can Google what those mean in terms of like HSV space. And I want to see whether or not we can decode some of those aspects of experience that we've always attributed to visual areas. So like, can we decode color from prefrontal cortex? Can we decode spatial frequency? Now that the algorithms are better, now that the decoding techniques are better, I want to show that that information is there. So we've done it with object categories, like I mentioned in my talk. I want to show that we can do it for these other dimensions. And then secondly, what I'm trying to do in some experiments right now is to use what's called neurofeedback to change neural representations in PFC. And I want to see if that can change your perceptual experience in a given moment. So there are techniques that we can use now where you can lie in a scanner, and using neurofeedback, I can change patterns of activity in certain parts of the brain over time. And what I want to demonstrate is a causal role for being able to change neural patterns in PFC and have that directly affect your perceptual experience in a given moment. Because I think what this area is really missing right now is a conclusive causal demonstration that if you change activity in PFC, it affects what you see in any given moment. We know from, say, surgeries on epileptic patients, where if we insert electrodes into the posterior areas of their brain and we actually stimulate those electrodes, it can cause visual hallucinations. So that's really informative and interesting from a scientific standpoint, because it provides evidence that parts of posterior areas can actually result in changing the conscious content that you experience. So there's a great paper in PNAS by, I think it's Gerwin Schulk and Nancy Canwisher, where they show that in epileptic patients, if you stimulate certain posterior areas, the people start hallucinating rainbows and hallucinating faces in objects. And that's a great demonstration that these areas play critical roles in what you see in a given moment. And what I would like to do is to use other neurofeedback techniques to try to show that if you change something about neurons in PFC, you're actually modifying the content. Because I think in this idea that PFC plays a critical role in conscious experience, it's those types of scientific demonstrations that would be most informative to demonstrate a role for this area. So that's where my research is going right now in the coming year. And if you keep an eye on my Google Scholar page or my lab website that I'll update throughout the coming year, hopefully you can stay abreast of the new results that I have to see what new things we're coming out with from the lab.
Excellent. Well, thank you so much. I would like to thank Dr. Odegaard for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June of 2018. You will receive an email from LabRoots letting you know when the webcast will be available for replay. Please share this announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you here again soon at LabRoots. Goodbye.